Hi there, thanks for the introduction. That saves me some seconds. And also, I'm going to talk to you today about some chemical compounds that we have designed in our lab at IRB Barcelona. And they can target a protein within the cell and use the cell's own recycling machinery to then target that cell and break it down, removing it from the cell. Now, our body is made up of many different types of proteins, and they can signal to each other in pathways to create an effect or a function in the cell, and rather like a domino effect. But when one of these proteins is overactive, or if there's too much of a particular protein, it can cause pathways to go out of control, and it can lead to diseases such as cancer. Now, one of the ways that the pharma industry can tackle this problem is by blocking the particular problematic protein with chemical inhibitors. They block its function and stop the downstream effects from taking place to try and get a hold on the disease. And what this looks like is we find an active part on the protein structure, the active site, shown as this lock. And we design chemical molecules that have the specific size and shape to come in to the and lock the protein's function by the active site. And there's over 150 molecules that act in this way, FDA-approved drugs, treating over 33 different types of cancers. As you know, many of these compounds have serious side effects, and people are still dying of cancer because they don't have a treatment for the particular stage of the cancer or for the particular type. So more work needs to be done to improve the situation. One of these ways could be to destroy the particular protein. So we work on P38 alpha. It's a protein that's important in the pathways that cause the formation of the tumor and also the spread throughout the body during metastasis. And we first find molecules that can specifically bind to this protein. And then we add on this second part linked to our molecule. Now this second part grabs an enzyme that's naturally found in the cells. And this is the enzyme that tags all the trash in the cell for destruction. By bringing it close to our protein, it causes it to label our protein as trash with an ubiquitin label. And then whenever the cell's recycling machinery comes along, this is the proteasome, this conical structure here, and it will recognize our protein as trash and will break it down and remove it from the cell. Okay, so we made a number of compounds with the two sides of different length linkers, different distances apart, until we found a perfect match. So, our compound finally had the right size and shape to initiate this destruction of the protein, and we then started focusing on the therapeutic benefit of our compound, different cancer cell lines, and we're now in vivo with it. So this does indeed have a stronger effect compared to the corresponding inhibitor molecules available in many cases. And you can apply this destruction of the protein at any time in the development of the cell or the organism without affecting the DNA or the RNA in the cell like genetic manipulation would in this case. So there are many other laboratories and companies that are also using this technology to find new targets, protein targets. And Arvinas has just released a phase 1 clinical trial using this technology in humans for the first time. So we're just that one step closer to actually getting to patients with cancer. And I need a little bit of help. I know this was...
yeah, I know, she's helping you.
So...
Hold the microphone.
This was a lot of information to take in probably, so I'd just like to summarize it now. Thought I'd sing a summary of all this information. I talked about a process using ubiquitination to induce the protease on causing protein degradation. This technology works, but it's still in its creation. About to go from testing labs to administering to patients. A future cancer therapy called protein degradation. Innovation was the mission of most medication. The low affinity and side effects are just two limitations. This is protein degradation. degradation. This is protein degradation. This is protein degradation. This is protein degradation.